Okay, good morning, everyone. Let's get started because we have a very tight schedule today. My great pleasure to welcome you to this session on current topics in molecular plant-microbe interactions that I'm co-chairing with my colleague Jane Leach here, of course, President of the International Society of Plant Pathology. And
our next speaker is Saskia HOGENHOUT from the John Innes Laboratory. And she's going to talk to us today about factors that influence the distribution of vector-borne parasites. Welcome.
Thank you. Thank you for that introduction. I'm really honored to give this talk here. It's very intimidating, this big hall, but I'll do my best. So I'm going to talk about phytoplasmas, which are transmitted by leafhoppers. These are bacteria, and they cause these really massive symptoms, very interesting symptoms. And that includes witch's broom and phyllodes. And phyllodes is actually the retrogression of flowers into leaf-like structures. And, well, as you know, there are several bacteria that are insect transmitted, and they all cause massive economic losses over the world. And Libribacter is one of them, and that's citrus greening disease in citrus in Florida, but also in South America. And it's destroying currently the citrus industry there. And these are transmitted by psyllids. And then there's Spiroplasma, which are transmitted by leafhoppers. And Spiroplasma conchylia is currently a massive issue, maize production in South America. Then phytoplasmas, which is where I work on, is also causing a lot of different diseases globally, including in lime trees. So lime is a big crop in the Middle East, and these phytoplasmas destroy the lime production industry in the Middle East. And then, of course, last but not least, Xylella fastidiosa. And Xylella fastidiosa has been an issue all over Americas for a long time. But in recent years, it causes the olive quick decline syndrome in Apulia, South Italy. And these are transmitted by sharpshooters and froghoppers. And my topic today is on phytoplasmas. And I'm interested because these phytoplasmas cause really massive symptoms, including like developmental phenotypes in plants. And phytoplasmas are molecules, they are soft-skinned organisms, and they don't have an outer cell wall. They're small bacteria, as you can see here. And what's really interesting is that they reside directly inside the cytoplasm of a host cell, including the host cell of a plant in the phloem, and also in an insect. And by doing so, they can secrete effectors directly into the cytoplasm. They don't need a type 3 secretion system. They only need a normal basic secretion system to deliver effectors into the host cell. And different molecules are defined, but phytoplasma is one of them. And they're also obligates. We cannot culture them, and they have a very reduced metabolism, so we just study these bacteria inside the host. So what they do is they increase, so they have these symptoms, and that also attracts the insect vector. So you have both massive symptoms, but also that the insect vector likes those symptoms and start colonizing the plants with symptoms better. And here you can see some different plant species that have symptoms, including witch's broom and also this phyllodes. And we call these zombie plants because the zombie plants are, they only there to help the bacteria propagate. The plants themselves are sterile, so they are not having progeny. So this is why we call them zombie plants. And we can actually improve, we have actually shown that the insects have increased fecundity, both in laboratory conditions as well as in field conditions. So we work on one of the phytoplasmas. There are many, many described. And this one is called Arstiellus phytoplasma witch's broom. And we like this one because it infects the model plant, because the phytoplasma itself cannot be cultured, so we want at least one model organism to use for our studies. And in this Arabidopsis thaliana, it also induces vicious broom and leaf-like flowers. And we wondered, okay, so how does the phytoplasma then induce this vicious broom, leaf-like flowers, and also increases the insect attraction to the plants and fecundity? So what we did is we sequenced the phytoplasma and we characterized a whole bunch of virulence vectors. And we call these SAPs, and these have nothing to do with the SAPs that were in the previous talk. These are called secreted Aspergillus vicious broom proteins, and that they are secreted in the sap of the plant. And there are actually two groups of effectors, one that are upregulated when the phytoplasma is in the plant, and the other one that when upregulated when the phytoplasma is in the insect vector. And for those in the plant, we have focused on 3 effectors called SAP5, SAP54, and SAP11. And what happens is that the phytoplasma is deposited in the phloem of the sieve elements, as you can see here by the leaf hopper. Then the phytoplasmas move between phloem cells through the sieve pores, and they secrete these effectors, including SAP11, SAP5, and SAP54. The phytoplasmas themselves stay in the phloem, but the effectors can migrate from the phloem to other tissues. So the effectors can move systemically and cause different symptoms. So we found out that SAP11, SAP5, and SAP54 do different things. SAP11 and SAP5 both induce this vicious broom and also increase the attractiveness and the fecundity of the insect vector. And SAP54 is inducing these leaf-like flowers and also increase the fecundity of the insect vectors. And today I'm going to say a bit more about SAP5. And SAP5, when you overexpress SAP5, you get this interesting phenotype in Arabidopsis thaliana, and you can see that on the right. And the SAP5 is inducing both more leaf production and more stem production at the same time, and causes these massive bushy symptoms, as you can see. And how it works. So we found out that via use to hybrid screens and other assays that SAP5 actually interact with multiple transcription factors. And these include SPL and GATAs. And there are many described in plants. There are many big families of transcription factors. And we found out that SAP5 actually interact with most of these, all the ones in red here in the slide. And it does that by interacting with the zinc finger domain of these transcription factors. And the zinc fingers are both, in both groups, they conserved. And even though the transcription factors are completely not related. So that's how the SAP5 interact with many. And what happens is that these SPLs and GATAs, they actually have a big role in the development of the plant. SPLs promotes the transition from juvenile to adult and reproductive phases in the plant. And by inhibiting that, you do get delay of development and more leaf production and younger plants. So plants stay young and they don't mature. The GATAs, on the other hand, are inhibiting branching. And by inhibiting that, you get actually more branching. So then you inhibit flowering, but the SAP5 is then promoting the production of stems. And then you get these kind of plants. And the zombie plants. And what's really interesting also is that during plant maturation, you get plants that accumulate defense compounds. So then, if that's the case, if SAP5 does its role, then you would expect there are less defense compounds generated. And this might affect the insect vector. And we tested this in both choice fecundity assays as well as no choice fecundity assays. And indeed, when in choice fecundity assays, you see that the leaf hopper prefers the SAP5 plants, and also in no choice fecundity assays. So the SAP5 does definitely increase the fecundity of the insect vector and the attraction. So then we wondered, okay, what's the mechanism? So what's the mechanism of how SEP5 degrades SPLs in GATAs? And so we find out that actually there's a susceptibility factor called RPN10. And RPN10 is a component of the yeast of the cis26S proteasome subunit, as you can see here. And it's a shuttle factor. So what it does is jacks components that has to be degraded to the 26 proteasome. And SEP5 interacts specifically with the VWA domain of this particular protein. And which is interesting, because normally that green area, the ubiquitin binding domains is binding. So SEP5 is binding to a unique part of this protein. And then we found out that actually SEP5 makes a connection between the SPLs, GATAs, and RPN10. And that leads to the degradation of SPLs and GATAs, as you can see here. As an example for GATA, but we have found this for all these different transcription factors. They're all degraded, and in an RPN10 dependent manner. So without RPN10, it's not degraded, as you can see here. But when we add RPN10, then you see degradation. So it's definitely RPN10 dependent. So this is what we see actually. So what happens is SEP5 forms a short circuit between two pathways. One is the plant developmental pathway that are involving SPLs and GATAs, and the other one is the protein degradation pathway. And so it connects the two, and then you get degradation of transcription factors. And so here you get then the short circuits. So short circuit, another short circuit. And SPL and 11 and 54 do the same thing, except that they degrade matchbox transcription factors and TCPs. So they also form short circuits between the plant developmental pathways and 26S proteasomes. And this is important because these symptoms will attract the phytoplasma insect factors. And also the plants degrade, or they mature less quickly. And so the factors will have more opportunities to acquire the phytoplasma. And there are other benefits, potential benefits for phytoplasma. Of course, there are more lethal perfectors, but it's also delayed plant aging and flowering. And most of the time, these Aspergillus phytoplasmas do infect annual plants. So when the annual plants die, their host or their house is gone. So they will benefit from delaying aging and plant death. And of course, when the plants do not flower, the nutrition is available perhaps for the phytoplasma, because the phytoplasmas do go really after zinc tissues. So instead of the nutrients being used for seed production, the phytoplasma may use them. So how can we apply this thing? So this is very interesting, but how can we apply these different aspects of SEP5? Turns out that we can actually translate the science. So we can reduce phytoplasma-induced symptoms in a broad range of crop trees. So we can actually find resistance. We can also use it to generate more attractive ornamentals. And we actually can also use it to develop drugs. And I will explain a little bit about each of these applications. One is to reduce phytoplasma-induced symptoms. Yeah, and so that's what I'm going to talk about first. And to realize why this might be working, you have to understand that the phytoplasma also colonize insects quite extensively. And you can see that in different tissues, phytoplasmas are in the gut cells, they replicate there, they migrate to the hemolymph, they infect salivary gland cells. And after they go after the salivary glands, and in the saliva, they transmit it to the plant and they cause symptoms there. So we wondered, okay, the SEP5 is going after SPLs and GATAs, but the GATA transcription factors are also very common in animals. So is actually SEP5 also modulating the insect factor? That's what we're wondering. So we looked at that, and it turns out that first of all, SEP5 does not bind the GATAs of insect factors, only the GATA transcription factors of plants. But it also does not bind this susceptibility factor, RPN10. And so we tested here and used the hybrid. So SAP5 binds, of course, the RPN10 protein of plants, but not the RPN10 protein of the insect vector, the leafhopper. And this is really interesting because RPN10 is super conserved. It's a very conserved protein in many organisms. And by comparing plant and animal RPN10s, we could actually identify certain regions that in plants RPN10 is involved in binding the SAP5, but apparently in animal RPN10 is not. So then we use that knowledge to make some mutations. And one of them is changing the GA in HS in the plant RPN10, called an M1 protein, and then also changing the GKG into K, and it's the M2 mutation. And then we found out that these do not bind SAP5. And then we take those mutants and introduce them in Arabidopsis thaliana. And so you can see here that if you introduce a wild type RPN10 back into a mutant of Arabidopsis thaliana, that in the introduction of SAP5 you get the symptoms. However, if we then introduce the mutant RPN10, which contains the mutations that inhibit the binding of SAP5, or insect RPN10, we see that the symptoms disappear. And this is also true in as of infected plants. So here, if you wild-type plants that are not transformed, and you can see that the phytoplasm is inducing these witch's broom symptoms. And this also happens when you do wild-type RPN10 that's not mutated. We just introduce it back into the plant, the mutant plant, and then you can see still the witch's broom symptoms. However, if we then introduce the mutant RPN10 that does not bind SAP5, the symptoms disappear. So basically we have plant resistance to the SAP5 effector. And so this is important because actually witch's broom is a huge issue, and in lime productions in the Middle East, but also in many other phytoplasmas cause witch's broom. So we are wondering, like, okay, so there's an RPN10 susceptibility factor in these trees. Can we change this RPN10 into a non-susceptible variant that does not bind SAP5, and then we might get rid of these massive symptoms. And this not only gets rid of the symptoms, maybe, but also affects the insect vector attraction, because as you can see here on the graph on the right, the phytoplasma, the symptomatic plants are very attractive to the insect vector. And these studies done in groves by collaborators in Oman, in Ali Al-Subdi and Abdullah Al-Sadi, and they did this in groves, in 3 different groves with different trees in each grove. And they put transparent traps inside the symptom and non-symptomatic plants, parts of the same tree. And you can see that the insects are much more attracted to the symptomatic parts of this tree. So if we make these symptoms disappear by perhaps inducing the mutation in RPN10 in these plants, then we might get not only rid of the symptoms, but also not stop the epidemics by not attracting the insect vector. And witch's broom symptoms are found in many different plant species, caused by different phytoplasmas. And here you can see all the phytoplasmas that have a SAP5 homologue. And in these phytoplasmas, they all induce these witch's broom symptoms. However, there is some evolution in SAP5 homologues, among SAP5 homologues, and that is that most of them actually bind both SPLs and GATAs, but some are evolved to only bind GATAs, and others only bind SPLs. But in the phytoplasmas that have these SAP5 homologues, these SAP5, they carry both homologues. So they have two SAP5 homologs, one that binds SPLs and one binds GATAs, except for apple proliferation phytoplasma. So, we also looked at which part of the SAP5 action is actually attractive to the insect vector. And it turns out that when the SAP5 degrades SPLs, the insect vector is more attracted to the plant. But if it only degrades GATAs, it doesn't. So it's really the SPL degradation that makes the plant more attractive to the insect vector. Now, a little bit about, just one slide about how we might use SAP5 for making nicer ornamentals. And for this, it's really important to know that actually quite a number of plants are actually infected by a stunting agent to make them more attractive. And you can see here in Poinsettia, the plant on the right is a bit more attractive than the plant on the left. And actually the stunting agent is often the phytoplasma. So maybe we can just get rid of this phytoplasma and instead use SAP5 to make the same kind of interesting plant. So, and the same for ficus. Ficus is also often, you can have beautiful different ficus varieties, and maybe SAP5 can be used to also make other varieties. And then and ornamental kales, for example. So maybe SAP5 can be useful in this way. Then the last part I would like to talk about is how we might use SAP5 to actually develop drugs. And and this is really important because SAP5 action is a new pathway that we found in cells. And this is the part, so it makes this short circuit by binding both the targets as well as a 26S proteasome shuttle vector. And this leads to ubiquitin-independent degradation. And this is actually new because this is a new pathway because the conventional pathway is always using ubiquitin. As you can see here, it requires E1, E2, and E3 ligases to ubiquitinate the target. And then the ubiquitinated target is actually degraded. And so with SAP5, we can completely bypass the ubiquitination pathway. And this is how we determine the structure of the complex. And so we can actually isolate the ternary complex by gel filtration, by combining all the three components. We can isolate the complex. So you can see here that SAP5 has a very interesting structure. It has two sides. One is the side the face that binds SPLs or GATAs, and the other side binds the RPN10. So it's like a molecular glue. And so this is pathway 1 and pathway 2, and then SAP5 forms the short circuit. And what's also really good is that actually SAP5 seems to be a bacterial subunit of the 26S proteasome. You can see here that it accommodates very nicely on the proteasome and does not cause steric hindrance. SAP5 binding to RPN10 the complex can bind without steric hindrance of the proteasome units. And what we also can do is we can actually degrade GFP with SAP5. So by fusing it to a GFP binding protein, we can now actually degrade GFP. So you can make it degrade other proteins. And SAP5 degrades protein via human 26 proteasomes. So this is isolated 26S proteasomes. If we add SAP5 and Arabidopsis thaliana VWA with the degraded target, we can actually use SAP5 to degrade other proteins with human proteasomes. So this is also interesting. And so to further develop SAP5 as a drug, because we can actually then use SAP5 to degrade maybe human proteins, we started up a company called Vector Med, and this with Sophien. Roberto Hofman, and that's funded by TSL Ventures. So what's next? So we have now characterized a whole bunch of phytoplasma vectors with interesting features. So what we want to do next, we want, of course, look for more vectors that have interesting abilities. And so we'll be looking for novel vector function with cellular path that new cellular pathways as well as short circuits. And we don't want to use the phytoplasma only. We also want to use insect vectors because these have actually not very much exploited yet. What do effectors of insects do, and can we use them to identify more interesting pathways in plants? And to do that, we also started a bit of a new program on insect vectors. And the first thing we did is to look to do a lot of genome sequencing and identify vectors. And so many sap-feeding insects have been characterized, as you can see here, but not sequenced so far. And so we did a whole big genome sequencing project for 30 aphid species. Also, we have whole genome sequence data of 12 froghoppers, spittlebug species that are actually transmitting Xylella as part of the Bridget project. And with the Caliber project that's led by Adrian Fox here, in the UK, we are also looking at psyllids, which are sap-feeding insects. For all of these, we also have population genomics data. So we are in a very good position now to look at effectors, find them, and see which effectors might be conserved among the sap-feeding insects, and see how we might be able to use them for all kinds of purposes. So with that, I would like to acknowledge my lab and collaborators. We have a really, really productive collaboration with our insectary, a very beautiful entomology facility at the John Innes Center that allows us to work with exotic insects, and also with collaborators at the TSL and Sophien KAMOUN and Abbas Makbul, and also and others that I've already mentioned. And then we also do the effector network and to further develop the SAP eraser technology using SAP5. I also would like to thank all my former lab members who have had various contributions to the work that I just described. And many of them have independent group leader positions all over the world, and especially Weijie Huang, who has done the SAP5 work and is now a project leader, independent project leader at the Chinese Academy of Sciences. Of course, the funding of the work. Then Jashveer Singh and John Chwin Liu and Vadim Merkin have really contributed to the SAP5 structure analysis. And last but not least, the zombie plant that I show today is drawn by my niece, Babette van Wijk. And Husam Pai, who is really a very talented illustrator at TSL, has developed the front page of the Cell paper that we published. So with that, thank you for your attention, and hope there is some time for questions. If not, then we might be able to tell you.
Thank you, Saskia, for that beautiful example of the translation of basic research to application of control of insects and pathogens. We have time for maybe one or two questions. We have many online, if there's someone live. If not, I'll take one from the online part. The first one would be, I had it right here. Fantastic work, Saskia.
Thank you.
How do you avoid degradation of SAP5 by the proteasome and overwhelming of the proteasome if you activate the proteasome-mediated degradation constitutively?
Sorry, I didn't understand the question quite. It's a bit...
Yeah, I'll repeat it.
Yeah.
How do you avoid degradation of SAP5 by the proteasome and overwhelming the proteasome if you activate the proteasome-mediated degradation constitutively?
Okay, yeah, it's a very good question. Actually, SAP5 is recycled. It's not degraded itself. So that's very interesting. It's Probably because RPN10 itself is also not degraded. So we think that the SAP5 interaction with the RPN10 prevents it from degradation. And that's really, really important because SAP5 can now be recycled to bind more targets. And also you don't need a lot of the protein. It's acts like enzymatically.
And one final question. Do you know how SAP proteins are secreted in the phytoplasma, from the phytoplasma to the phloem? Is the production and secretion of SAP proteins plant-specific?
Yeah, so phytoplasmas just have a general SEC-dependent pathway. So it just the SAP effectors have a normal general signal peptide. The signal peptide is cleaved off, and then the mature protein is then degraded into the secreted into the phloem via the SEC-dependent pathway. So it's just a general secretion pathway that any organism has. And the reason is that the phytoplasmas reside directly in the cytoplasm of a plant. It is not requiring any sophisticated type 3 secretion system to get our effectors into a plant cell.
Thank you, Saskia.
Lots of progress, but still a lot to figure out. That's a great closing statement. And I really encourage you, Shenghan, and the other speakers to interact with the questions in the app. So thank you all for your patience and for joining the session. And thank you, Jane, for co-chairing. And we'll enjoy your coffee break.